Cancer is something we touched on previously in our slides about mitosis. So as a reminder, cancer develops when normal cells become unresponsive to the stop and go signals that normally control mitosis. Some cells divide pretty fast. Your skin cells, your hair cells, they divide pretty fast. Your brain cells divide very slowly, if at all. There's some debate about that right now. So different cell types need to regulate how fast they go through cell division. And as you know from the other videos in this playlist, the regulation of cellular activities is always done by controlling which genes are transcribed, which genes are translated, how often, how fast, all that kind of thing. So when we say that these cells have become unresponsive to stop-go signals, it means that there's been some sort of mutation to their genes, which normally should either prevent cancer or encourage cell division. There's been some sort of mutation that causes the dynamics to change, and so one cell with a mutation inside of a tissue starts dividing and dividing and dividing and dividing, and eventually you get this mass of abnormal mutant cells. Typically, because they divide so fast, they don't stop for very long for interphase in between divisions, so they will become a lot more dense than the surrounding tissue, because interphase is when they put on some nice mask or the cytoplasm, that kind of thing. So cancer is a disease of the cell cycle, but it relates back to our conversations about cell gene expression control. So just as a reminder, throughout cell division, there's certain checkpoints that have to be made. One of them, a really important one, happens in metaphase, where it verifies that all of the microtubules of the mitotic spindle have actually attached to the chromosomes before it's allowed to proceed to anaphase. If they have, right, there's proteins that'll verify that, and then they'll send signals, and we'll make sure that the whole process goes ahead. But you can imagine how if, let's say, the cell cycle, the attachment wasn't perfect, and we allowed it to continue anyway, well, we could get uneven separations of chromosomes, or of chromatids here, which would result in even more mutated cells. Another checkpoint is in G1 of interphase, and just verifies that the cell has put on enough mass, has developed all of the correct organelles and tissues and parts, well, not tissues, and parts that it needs in order to proceed to the next phase. So what we're talking about is mutations of specific kinds of genes that are meant to control cell division. Cancer results from mutations in genes that control cell division. Cancer cells, which divide uncontrollably, result from mutations in genes whose protein products affect the cell cycle. A mutation can change a proto-oncogene. Now, let's break down this word. Proto means before, it means pre, it means it's something that's going to become something else. Onco, O-N-C-O, a person who studies cancer is called an oncologist. The department they work in is oncology. So oncology is the study of cancer. That should tell you that this prefix onco means cancer. And then gene, section of DNA that codes for certain proteins. So what is a proto-oncogene? It's any normal gene inside your genome, genes that are supposed to be there, that have a role in either speeding up cell division or slowing down cell division, or have a role in one of those checkpoints I was just talking about. It's going to stop cell division until this criteria is met. Anything that is meant to control the cell cycle, if it mutates, can become what we call an oncogene, which is a cancer-causing gene, a cancer-causing mutation, essentially. So all of the genes which affect the cell cycle could be characterized as proto-oncogenes in this framework, because with a mutation, they can then become a cancer-causing gene, which we call an oncogene. There's also another group of genes which are called tumor suppressor genes, exactly what they sound like. They're meant to detect when cells are tumorous, when they become cancerous, and either shut down that cell or in other ways repair the damage to the DNA. They're meant to prevent the formation of tumors. But if you get mutations in those tumor suppressor genes, then that can also lead to cancer. Okay, so let's say that this up here is a section of DNA with a gene that helps regulate the cell cycle. That's what we call a proto-oncogene. And a proto-oncogene, again, is a working gene. It's doing what it's supposed to be doing. It's just its job is controlling cell cycle. With a mutation within the gene will turn it into an oncogene. And let's say it's supposed to make a protein that stimulates cell division, right? Its job is to say, oh, hey, it's time to divide. The mutated version of this gene might make a hyperactive growth-stimulating protein, which means the cell is going to divide more and more and more and more because suddenly the protein it normally makes is become supercharged because of little changes inside of that gene. So that's the kind of thing we're talking about. This is meant to stimulate cell division, but with a mutation, it might go too far and become a cancer-causing gene. You could also get a kind of mutation, we call them duplications, where entire sections of the chromosome get copied by accident. It's kind of like, you guys remember that that DNA replication is accomplished by DNA polymerase, and it starts at certain points and then continues on its way through. So it's kind of like if it skipped on a record and it replayed the same section over again. It happens when it gets double copied and you get two sections of a gene instead of the one copy you were supposed to get. So if that happens, if we accidentally get multiple copies of the gene, then that normal growth stimulating protein is going to be created. It's not the hypercharged version, but if every single one of these genes is being translated, we're going to get more and more and more of that normal growth hormone, and that can also lead to excessive division, so that can cause cancer as well. And then we could also get a mutated promoter region of the DNA, which causes the transcription factors to attach to it more often, more transcription. So we still have just one copy of the gene. The mutation isn't in the gene, it's in the promoter, but it's still being made more and more and more and more, and you have the same thing as you had in the situation with multiple genes. More of this growth stimulating hormone is being produced, and that can cause cancer. So these are three different ways that a mutation could take a normal functioning proto-oncogene and create cancer as a result. Tumor suppressor genes are genes that create proteins that inhibit excessive growth. So this is a normal growth inhibiting protein, and it prevents cells from dividing out of control. But if we get a mutation in that tumor suppressor gene, then it's not inhibiting any cell division, which can then cause these cells to divide over and over and over again, creating, again, cancer. So proto-oncogenes and tumor suppressor genes can get mutations which then lead to cancer. Sometimes a single mutation isn't enough. You need multiple mutations in order to get cancer. But the more it replicates, the more mutations you're going to see. Every time DNA is copied, every time mitosis happens, or every time we have babies, you get DNA being copied, and there's a chance of mutations popping into the genome, slight spell check errors in the copying of the DNA. And some of them are caught by our cell's internal machinery. We basically have a spell checking system inside of our cells, but some of them make their way through. Every time DNA is replicated, you can get more and more mutations. And maybe the first couple of mutations 1, 2, and 3, are harmless. They don't actually do anything by themselves. But that fourth mutation changes things just enough that now the protein that they're creating is substantially different, and it creates a cancer cell as a result. So this happens quite frequently, where you have multiple mutations, and it's that last one that really pushes it over the edge. So this is an example of how multiple mutations could lead to a really malignant tumor. We have the wall of a colon with a particular tissue type in it. A single mutation could cause an oncogene to become activated, and that would increase cell division. Now, if we have increased cell division, tumor suppressor genes are in place to prevent it from getting out of control. So even with this oncogene active here, we still have the chance for tumor suppressor genes to shut that down. But if there's a second mutation that's in the tumor suppressor gene, that will allow the cell division to continue, and you get the growth of a polyp. And now this is not necessarily cancer. This could be, say, a benign tumor. But another tumor suppressor gene gets a mutation in it, and suddenly you get the growth of a malignant tumor. There's no regulation of the cell division now. It just keeps growing, growing, and growing. Now, the more this grows, parts of it can break off and develop more tumor polyps, more tumors elsewhere in the colon. And if it gets into your other systems, that's what we call metastasis, where a tumor in one location, a cancer in one location in the body, starts spreading to other locations in the body. That's stage 4. That's really bad when that happens. Now, I said that mutations occur naturally as a process from cell division and from DNA replication, but there are certain mutagens you can take into your body which will increase the likelihood of mutations to occur, and some of them will end up causing cancer. So this is why it's important to try to put on sunblock when you go outside, because UV radiation increases the mutation rate of your skin cells. It breaks your DNA and then has to repair itself. So that is a mutagen. Smoking cigarettes, there's many chemicals inside of cigarettes that cause mutations, and that can cause cancer as well. So living a healthy life really increases your chances to live a long, healthy, prosperous life cancer-free. It's not an absolute guarantee. I'm not saying it's an absolute guarantee, but it does increase your odds. Faulty proteins can interfere with normal signal transduction pathways. So we've just seen how some faulty proteins, some faulty genes can lead to cancer. But here's some other problems that can happen. So signal transduction pathways lead to the synthesis of proteins that influence the cell cycle. Many proto-oncogenes and tumor suppressor genes code for proteins active in signal transduction pathways regulating cell division. So I told you that the way that cells differentiate is by talking to each other, cell-to-cell communication. And that's how they determine, oh, I'm going to be a muscle cell, I'm going to be a bone cell. But once they get that identity, cells don't stop talking to each other. They're constantly talking to each other. If one cell becomes cancerous, the cells around it will start sending signals that are meant to cause actions that will get that cell to stop dividing and dividing. We call that contact inhibition, when the other cells around it are basically saying, hey man, you're dividing too much, calm down, you've gone all screwy here. It's called contact inhibition when we use cell-to-cell communication to prevent cancer. But mutations can occur in the genes that govern those signal transduction pathways, which then make the cell basically deaf to contact inhibition. It has no contact inhibition present. Two different examples here. We have the normal situation where some growth factor would come in and attach to the target cell. Now it's a growth factor, so this is meant to increase cell division. And that receptor will release this relay protein, which is a product of a gene we call the RAS gene. That's what's going to be mutated in a moment. The relay proteins do what they do. They have a domino effect where they keep activating each other, and then they activate the transcription factors, which then cause certain genes to be transcribed and translated, and then that creates a protein which stimulates cell division, creating normal cell division here, right? So this is a situation where cells next to the cell that we're looking at are saying, hey, you need to divide more, all right? And it says, okay, I will divide more, and this is the process by which that division will be triggered. But when we have a mutation, then the RAS protein, the RAS gene, becomes hyperactivated even when there's no signal telling it to become activated. So the cells aren't saying anything next to this one. They didn't say start growing, but it just starts dividing and dividing and dividing anyway. So this is one of the ways that signal transduction pathways can go awry and lead to cancer. Another example, this is where contact inhibition really comes up. We have a growth inhibiting factor. So some cell next to this one said, hey, stop dividing. I don't want you to do any more divisions. We have all the cells we need right now. And in the normal process, the receptor will receive it. It will go through a series of relay proteins, and they will activate this molecule, which is called P53. It's a tumor suppressor molecule. It's a growth suppressor molecule. And then the transcription factors will be activated, which then create another protein which inhibits cell division. Okay, so I know this looks like a whole complicated series of steps, but all this is really saying is other cells are telling this cell to stop growing. It receives the signal, goes through a chain of proteins, and then it creates another protein that stops the cell division. Signal received, consequence occurs. But if there's a mutation in the cell of this critical P53 protein that's meant to activate transcription, then what will happen is it will not ever turn on that tumor suppressor gene. It will no longer activate it, which means that we will not have that protein created, which decreases cell division. And as a result, we'll get increased cell division. So even though the other cells are saying, stop growing, stop growing, stop growing, stop growing, this cell just keeps growing because it has lost the ability to interpret that signal. It's lost contact inhibition. Mutations of P53 are one of the most common causes of cancer. Okay, so this is a review chart of a lot of the things that we've talked about in this video series so far. Gene regulation is how genes are turned on and off in various cell types. Prokaryotic genes are often grouped into something called an operon. Operons are controlled by a protein called, in its active form, it binds to blank. And here's a clue. They've given you a little picture of B and D right here. Let's take a look at A. Blank is a normal gene that can be mutated into an oncogene, and it can cause blank. Then we have in eukaryotes, gene regulation may involve blank, which occurs in female mammals, blank, which involves proteins that promote transcription, and blank, which can produce multiple kinds of mRNA per gene. So hopefully you've been watching all of these videos, and some of these are sounding familiar. I'll give you a moment to log in your answers. Okay, hopefully you paused the video and tried it yourself. Let's start with A. A normal gene that can be mutated into an oncogene. That's a proto-oncogene, right? A pre-cancer gene. What can be mutated into a cancer gene? A pre-cancer gene. A proto-oncogene can become an oncogene. And that can cause cancer. B. Operons are controlled by a protein called the repressor. This is the repressor right here. In active form, the repressor binds to, remember prog, P-R-O-G, so here's the promoter here, and the repressor binds to the operator. So this is the operator, D is the operator. Gene regulation in eukaryotes may involve blank, which occurs in female mammals. X inactivation, that's where randomly one X chromosome in each cell will be basically the DNA will be wound so tightly around the histone proteins that the whole thing becomes heterochromatin, and you can't use any of the DNA there. So we call that X inactivation, where one of the X chromosomes gets basically crumpled up and becomes what we call a bar body. That little crumpled up X chromosome is a bar body. But E is X inactivation. Gene regulation in eukaryotes may involve F, which are proteins that promote transcription factors. Remember those tissue-specific proteins which promote transcription? Those are transcription factors. And activators are the specific kind of transcription factors that attach to the enhancers, and that really ramps up transcription. Gene regulation in eukaryotes may involve blank, which can produce multiple kinds of mRNA per gene, alternative gene splicing. Remember when we had introns and exons and we could decide which exons we were going to leave in to get different kinds of protein from the same gene? That's called alternative gene splicing.